Yep, we're good to go.
All right, so hi everybody. Thank you for coming to our fourth installment of the Biological Stoichiometry seminar series. Today we're going to be still in the water realm, but getting the first time in which we were getting a little salty and dealing with oceans. So today's speaker is Dr. Cathy Garcia, and she's currently a postdoc at the University of Hawaii. And she's going to be focusing this talk on her PhD work, which was done at the University of California, Irvine, and in coordination with the Bigelow Lab and also with Princeton University. And with that, I will turn it over to Dr. Garcia, and welcome everyone.
Hey, I just wanted to say thank you for allowing me to give this talk. This is actually a really enjoyable chapter of my PhD. I had collaborators from the Bigelow Laboratory for Ocean Sciences and also George Hagstrom from Simon Levine's group at Princeton University. So I guess this is the fourth talk in this series. Apologize if you guys see names kind of popping up on my screen. I don't know how to make that go away with the screen share. All right. Okay. So again, my study system is going to be the near surface, subtropical, and tropical open ocean. This will be the latitudes maybe about 30 north to 30 south. In this area, when we're talking about the typical environmental factors that limit phytoplankton growth, nutrients, light, and temperature, we're not really thinking about light limitation here. Most of these samples are going to be in the mixed layer, so in the top 10 meters at least. And really what we're looking at is gradients in nutrient supply and gradients in temperature across the surface ocean. And both of these are well known to limit phytoplankton growth. But the question is, how are these environmental factors, nutrients and temperature, going to affect phytoplankton growth, kind of looking forward into the future? So here I'm kind of, it's a little bit of a cartoon of two different scenarios, where on the left is the modern ocean, where in the ocean nutrient supply is coming from deep for the most part. There are also other sources from nitrogen fixation and dust sources as well. But for the most part, the macronutrients are kind of being upwelled from deep. And in a warming ocean, you have this effect where that surface layer becomes increasingly stratified and separated from the deeper layer. So the question is, in these low latitudes, what is the effect of not just temperature, but also potentially decreased nutrients to the mixed layer going to be on these phytoplankton? So I wanted to pull this up since the IPCC report has gotten a lot of focus recently. This is a figure from a special report in 2019 from a study by, I'm going to say this wrong, Lafcader et al. In 2015. And what it's showing here are in an assemble of different CESM models here. That's what all the different gray lines are. And you can see the effect of nutrients, light, and temperature on limiting net primary production among these different ensemble runs. And one of the main things to take away from this is that if you can see in the left panel over here with nutrients, you can see there's quite some disagreement in this low latitude region. I'm kind of highlighting in orange here, where a couple of the ensembles with this positive, it would mean decreased limitation on net primary production versus increased limitation on net primary production. So there's some disagreement here, even though overall they tend to kind of average out towards decreased nutrients would be limiting net primary production. But then on the other side of that, you have this opposite effect of temperature, where as you increase temperature, as that might have an effect on the enzyme kinetics and growth rates, that may be predicted to increase net primary productivity among these different ensemble models. So there's really this question going forward is, in this low latitude region, what is the effect of the combination of these factors in the surface where light shouldn't be limiting, but you have decreased nutrient supply to the surface, but increased temperature. So a lot of the work that I'll be focusing on is how to tease apart these two factors, because nutrient supply and temperature often are very, very well correlated in the ocean. Typically, when you look at these two factors at a global level, you have increasing temperature is associated with decreasing nutrients. And a lot of people kind of use these interchangeably when modeling phytoplankton growth. But what I'm going to be describing phytoplankton, but in reality, what I'm measuring is particulate organic matter. So a caveat here is POM, particulate organic matter, includes phytoplankton, heterotrophic bacteria, dead organic matter, the detrital material, really anything that's going to be caught onto a GFF filter, which is typically how these measurements are made. So all of the material for these POM carbon, nitrogen, and phosphorus observations are collected onto that filter. And then this is only going to be kind of like the smaller fraction, so under 30 microns. So I just wanted to put that out there. So historically, when looking at the ratio of these concentrations of the different elements of carbon to nitrogen to phosphorus, the seminal study by Redfield in 1934 kind of compared the ratio of these nutrients. So the nutrients in seawater, you can see in this right-hand panel. Let's see. Let me just minimize this real quick. I don't know if this is affecting. Okay, so if you guys can see on the right-hand plot, there's this very tight relationship between nitrate and phosphate where they all kind of fall along this line. And if on the left, if you plot observations of surface particulate organic nitrogen with particulate organic phosphorus, you see they also roughly fall on this 1-to-1 line, not 1-to-1, roughly 16-to-1. So based on this, Alfred Redfield came up with there you go, this really key theory that there's balanced proportions between phytoplankton nutrient uptake, and then as those particles sink in the ocean, they degrade and they're remineralized at these constant ratios of 16-to-1. But work by my advisor, Adam Martini, he compiled the surface observations of carbon, nitrogen, and phosphorus. And in the study, he found that there's actually systematic latitudinal variation among these ratios. This is a more recent compilation just of recent cruises in the Martini lab. And you can see the systematic variation where as we go from 60 degrees north latitude, there's an increase in the particulate organic C to P ratio as you get into the subtropical region, which is going to be warmer temperatures and low nutrients. There's a decrease in the ratio systematically across all these different ocean basins, Pacific, Atlantic, and Indian Ocean, as you get into the equatorial region of higher nutrient supply. But then there's a mirrored increase into the southern subtropical region. And again, a decrease in these ratios into the cold, high nutrient polar regions. So a lot of this kind of begs the initial research question, are in this particular area of the low latitudes, I'm interested in what are the dominant drivers of low latitudinal variation in these particulate C to N to P ratios. So a lot of work has already been done on these different environmental drivers. And here I'm going to highlight 3 of them. So one of them is the effect of temperature on these ratios. So through translation efficiency, it's predicted that as temperature increases, the phytoplankton they have increased efficiency and they need less phosphorus-rich ribosomes to grow at the same rate at elevated temperatures. So this results in a lower overall phosphorus quota per carbon molecule for the particulate organic matter. Now, as well, we have this kind of similar prediction for nutrient stress. And you might see me going back and forth between nutrient stress, nutrient availability. Really what I'm implying here is that as you have a decreased nutrient availability of these macronutrients, phosphate and nitrate, to the surface ocean, the cells exhibit nutrient stress and they adapt in different ways by decreasing their cell quotas for nitrogen and phosphorus. So again, this is going to lead to a predicted increase in C to P. And also the last one we sort of tested in one of these Indian Ocean studies is what is the effect of simply cell size and community composition. In Arrigo's paper in 2005, he kind of talked about different lifestyle strategies, a bloomer, survivalist, where small phytoplankton are kind of optimized to have lower N and P quotas. So again, a larger C to P versus larger phytoplankton are going to be able to kind of have luxury storage and be able to increase their internal cellular quotas. So the thing to notice here is that all of these are typically co-vary, and it's hard to tease apart these different factors. And if we're kind of looking on to the future warming scenarios, this is a nice kind of compilation study by Tanioki and Matsumoto last year. And they looked at these different environmental drivers of nutrient concentrations, increases in light and increases in warming. And what you can see is the combined effect of all these future changes, again, are predicted increases in the C to P and C to N ratio. So it kind of begs the question, how do these effects interact and how do we kind of tease them apart to see what the dominant drivers are? So one of the ways to do that is to use a trait model based approach. So this is a study by Alison Moreno, George Hagstrom et al. From 2018. George Hagstrom was also a collaboration for the trait model in the study that I'll be talking about going forward. But basically, they divided the phytoplankton cell into these different compartments. So for light harvesting, biosynthesis, storage, and went kind of modeled across different environmental conditions of light, temperature, and nutrients. And based on the cell quota for these different compartments within the cell, whatever one had the optimal fitness, we would calculate the C to N to P ratio for that. So from there, they could see what the effect of these different environmental drivers were across low latitude conditions in the subtropical and tropical global ocean. And generally what they saw for the interaction between temperature and phosphate on resource allocation was that, so phosphate had a very strong effect on the cell radius and P storage allocation. You can see that in these top two panels here, where as phosphate increases, you can see a strong effect on these two variables, but not much of an effect of temperature. Versus on the bottom, what Moreno and Hagstrom et al. Found was that at higher phosphate concentrations for biosynthesis and photosynthesis, when replete nutrients, there's an abundance of phosphate, temperature has a stronger effect on these two compartments. But once you start getting into phosphate limited conditions, there begins to be this interactive effect. So again, there's an interactive effect at the low phosphate concentration in these subtropical gyres. But the question is, which one is the stronger driver in kind of driving these particulate C to N to P ratios? So again, here you can just see the combined effect of these two variables at a set light level. You can see that the combined effect of phosphate and temperature leads to this interaction, and you have the highest C to P at low phosphate, sorry if you see my cat, and high temperature. To kind of test this theory, we chose a case study in the Indian Ocean. These are everybody who was kind of a part of that. Steve Bear, Sarah Ruschenberg, and Nathan Garcia on the right there. So again, trying to hold back my cat. So this is just a quick picture of our sample rig right here. Here you go. Sorry. Pet bombing. So we collected everything in triplicate from the underway sampling system from about approximately 5 meters deep. And what we found, the reason we chose the Indian Ocean is that it's an ideal area to test this hypothesis, because globally we find that temperature and nutrient supply are strongly correlated. So you can't really tease apart these different effects of temperature and nutrient stress and cell size. But in the Indian Ocean, nutrient supply and temperature are negatively correlated. And part of the reason for that is that you have, unlike the Atlantic and the Pacific Ocean basins, the Indian Ocean is landlocked to the north. So it tends to trap heat much more strongly, and you have this strong increase in temperature as you go north along this transect. But correspondingly, once we go south into the South Indian Ocean gyre, we have a decrease in nutrient supply. So we can tease apart these factors. So the key takeaway from this case study was that nutrient supply did show the strongest predictive value for C to N to P. So what I mean by that is as we went north, so this is the Southern Indian Ocean gyre. As we went north, we saw a decrease in the ratios of C to P, C to N, and N to P of particulate organic matter. But that was followed by an increase in temperature. So we would have expected, if temperature was a driving factor for changes in these ratios, that C to P would be higher as we went north into the Bay of Bengal. Instead, we didn't see that. What we did find is we looked at the, sorry, you guys can see again, another cat bombing. We had the lowest nutrients, the deepest nutricline depth, which I'm using as a proxy for nutrient supply in the South Indian Ocean gyre, which corresponded to the what we're calling nutrient limitation and the higher C to P, C to N, and N to P ratios. In biomass, even though there was an overall increase in biomass, the proportions of cyanobacteria to heterotrophic bacteria was approximately 50-50 over the course of the transect. So the composition of these different groups at this level using flow cytometry did not change over the course of the transect. And we did model the relative changes, and we did not find either temperature or biomass composition could explain the changes in C to P, yet nutrient supply as modeled by nutricline depth was able to explain the changes. And also just, again, going back to when we look at the global relationship, between C to P and nutricline depth in both the Indian Ocean in red and the global relationship both have this positive correlation. So as nutricline depth deepens, nutrient supply is implied to be lower, increasing the C to P. Versus when we compare that to temperature on the global scale, there's an inverse relationship here, kind of at least in leading us to the conclusion that in these low latitude areas, when nutrient supply is limiting, nutrient supply seems to be a more important driver of changes in particulate C to N to P ratios than temperature, at least in these low latitude regions. So from this, we wanted to just test that trait model and see how well the predictions of C to P came out using satellite PAR, so light, temperature from observations, and phosphate observations. What we found was actually there was a poor correlation between the observations of C to P. That's the axis shown on the right. So the observations are in black, where you can see that, and this is latitude, You can see that decrease in ratio as we move north into hot, warmer waters, but we see that the cellular trait model using these observations, either using straight the P observations or a proxy for phosphate, we can see that the trait model predicts an increase in C to P. And there's really two things driving this. One is the effect of temperature on this initial model. An increase in temperature would predict higher C to P ratios, but also we actually found that the Indian Ocean is actually quite interesting when it comes to surface phosphate, although I'm not sure if this name is blocking it. What you can see here, this is nitrate on the top. See if I can move this. Anyway, so this is nitrate on the top here and phosphate on the bottom. Even though both of them show this very deepening of the nutricline in the South Indian Ocean gyre, what we have here is phosphate concentrations at the surface where we're collecting these particulate organic matter observations are higher in the Southern Indian Ocean subtropical gyre and decrease as you go into the Bay of Bengal. So we also have this effect where surface phosphate appears to be less limiting, or appears to be more limiting in the Bay of Bengal, driving a prediction of increased C to P. So we really have this question of which nutrient is limiting in the Southern Indian Ocean gyre, causing this increase in C to P ratio. So we kind of come back to this trait model here. This is just adapted from that schematic I showed earlier, where you have these different compartments for biosynthesis, photosynthesis, and the nutrient uptake. What I wanted to show here was that the nutrient input for the original trait model is just using phosphate as a nutrient, and it is diffusion limited, the nutrient uptake. So we wanted to test this hypothesis where if there was a study by Twining et al. Showing I think the study is like a mosaic of nutrient limitation in the Indian Ocean, whereas there's this shift between nitrogen, iron, and phosphorus stress depending on where you are along that transect. So we wanted to add nitrate into the model. There's some studies by, I think, Nathan Garcia and Juan Bonacella, among others, that look at the effect of nutrient uptake transporters on the phytoplankton cell. So nutrient uptake transporters are an N limited portion of the cell. So we wanted to incorporate this into the model where the uptake of these nutrients is going to be limited based on the N availability. But there's a slight problem here. It turned out that nitrate was actually below detection for much of this transect. So we have this hypothesis, but we're not actually able to test it using the observations. So this kind of goes to the part where we're starting to combine different methods of the oceanographic observations, the trait model, and now trying to come up with another way to imply nutrient concentrations. So there's this hidden problem to trying to use this alternative nutrient nitrate, and that's that it's below detection limit of common assays. And this is actually a common problem unless you're using a high sensitivity phosphate assay called Magic SRP. Phosphate may also be below detection at many of these stations. As well as when we're looking at nutrients, in reality, nutrients, it means much more than nitrite, nitrate, phosphate. It can mean ammonia, dissolved organic nitrogen and phosphorus, nitrogen fixation, dust inputs, and inputs don't just come from the deep. They advect from horizontally. There's recycling. There's isopycnal mixing of nutrients. So really, how do we quantify all this variation in nutrient supply? One way that we thought of doing it is using the underlying taxonomic and genomic diversity. So what we take here is we're looking at the metagenome. So what that means is all the genes in a sample of water from the microbial community. and we're looking at the adaptation of these different samples. So we're looking at the assumption that the taxa that are present in the metagenome are the metagenomes present are the taxa that are best suited to their environment. So from this, instead of just having nutrient concentrations, we actually take we developed a nutrient stress index instead. So I'm going to talk a little bit about how we developed that. So some of the underlying assumptions for this, going into this, was that there's this underlying ecotype diversity in both the Indian Ocean transect shown by Larkin et al. In 2020, but also in these other transects where we had both DNA samples and particulate organic matter samples in the Pacific and the Atlantic Ocean, shown by Kent et al. In 2018. And by ecotype diversity, there's this nice figure that Alyssa Kent showed in her paper. So Synechococcus and Prochlorococcus, when you kind of increase the scale of diversity, if you kind of see it as a sliding scale, initially these clades tend to split off between low temperature and high temperature clades here. And there's kind of a scale of light followed by temperature, and then perhaps followed by high and this kind of high temperature, low iron. So you can see here in Prochlorococcus, it's a little bit easier. We have first, as you increase the diversity scale, you have these low light splitting off from the highlight ecotypes. Then as you further go along that diversity scale, you start seeing separation based on temperature. And as you go further along, you start seeing separation based on nutrients. So kind of as you increase whatever stress level there is along a gradient, whether it be light, temperature, or iron, we expect these ecotypes to be present in the metagenomes. So that is what we found. We found these ecotypes present in the Pacific, Indian, and Atlantic Ocean. Primarily, we found highlight ecotypes present in the, here, this would be the Indian Ocean transect, is really just all dominated by this highlight 2 ecotype. But what we see here is that some of those, if you see this, this is highlight 3-4 for Prochlorococcus. If you go back, so this highlight 3-4 is this high temperature, low iron clade. So the presence of that high temperature, low iron clade in green in the Pacific Ocean is indicating ecotype Prochlorococcus that is adapted to high temperature and low iron. And there's lots of support for this. The equatorial upwelling area in the Pacific Ocean is often thought of as a high nutrient, low chlorophyll area, hypothetically driven by reduced iron supply to this region. And we see kind of a corresponding increase in ecotypes associated with these H and LC areas in the Synechococcus ecotypes as well. So from this, we wanted to develop this nutrient stress index. So some key assumptions here are that we're assuming that the relative gene frequency, so by that we're looking at the genes for different kind of like nutrient assimilation pathways, whether related to nitrate, nitrite, ammonia, urea, assimilation within the cell. We're assuming that the relative frequency of each of these gene copies, that the gene coverage of this gene, when we normalize it to the median coverage in that sample, we expect that an increase in the relative gene frequency represents a cellular investment in nutrient assimilation genes. And the reason for this is that these picophytoplankton, so Prochlorococcus and Synechococcus, you can kind of see here on the top the schematic from Trichodesmium to dinoflagellates to diatoms, coccolithophores to these picophytoplankton. You can see that there's a size scaling here. So the smallest cells, Prochlorococcus and Synechococcus, they streamline their genomes so that it's really a presence or absence of genes that they need or they do not. And there's this really great, this wonderful figure. I think Elise Larkin made this, that really shows this for the different genomes that we map these different samples to. So over here on the right are the different representative genomes of Prochlorococcus here. And what you can see here is when the square is highlighted in black, that's going to be the presence of these genes associated here. These genes are all associated with nitrogen assimilation. These genes are all associated with phosphorus in blue, and these genes are all associated with iron. So what you can see is a lot of them, you can see that some of these clades have these gene copies, whereas other representative genomes do not. So when we're mapping these metagenomes to these reference genomes, there's really just a absence of these genes, which is how when there's an increase in the frequency, it's representing an adaptation to nutrient stress at these different stations. So this is just showing kind of like a heat map for these three elements in Atlantic, Pacific, and Indian Ocean. So what you're seeing here as a deepening of the color means an increased relative frequency, and we're interpreting that as increased adaptation and investment in kind of like acquiring those different nutrient sources. So what we see here is that for nitrogen, we see in the temperate ocean, there seems to be, as we expect, there's high supply of nitrate and nitrite. So there's little investment in these different, in all of the gene frequencies here. But you can see as you go to the subtropical North Atlantic, there's an increase in the frequency showing nitrogen limitation, as well in the tropical North Pacific. And basically throughout the Indian Ocean, we see evidence for nitrogen stress here. But we do not see it in the equatorial Pacific region. Again, this is high nutrient, low chlorophyll. When we go to phosphorus limitation, we see supporting a lot of work in the literature indicates phosphorus limitation in the subtropical North Atlantic. So we're seeing both nitrogen and phosphorus stress, evidence for that in the subtropical North Atlantic stations. We're not seeing any evidence for phosphorus limitation for the most part in the Pacific. In that Pacific transect, but we are seeing it in that Bay of Bengal area. So again, if you remember, I showed you that transect where we saw an increase in surface phosphate into the subtropical South Indian gyre, but a deepening of the nutricline. So there's these different nutrient proxies, but if we look at the genomic scale, we can see evidence for widespread nitrogen limitation in this Eastern Indian Ocean transect, but phosphorus limitation specifically in the Bay of Bengal. And again, we especially see evidence for iron limitation in that HNLC area of the equatorial Pacific Ocean. And that's kind of highlighted here. So how do we quantify this? Again, we were looking at those relative frequencies, and the way to kind of see this in a 2D schematic, we looked at the PCA plots to look at how much of the variance could be explained using principal component 1 and principal component 2. So from that, you can see kind of these, along this axis, you can see phosphorus limitation, then coming around to nitrogen limitation, and then iron limitation. So from that, we use the PCA along the first axis to develop this stress index. And we validated it against phosphate and nitrate concentrations along this research cruise. Just check the time. And what you can see here is that, again, anything highlighted in red is going to be from a high sensitivity assay. So all of these observations here are just at the detection limit of a standard dissolved inorganic phosphate assay. But basically what you see is that as this PCA gene contribution has a inverse relationship with the increase in phosphate molecules. So it was really nice validation here that we are able to see this evidence for this increase in surface phosphate using this nutrient stress index. Again, for nitrate, as I mentioned, pretty much all of the nitrate values were below detection for a lot of these cruises. There are low-level nitrogen assays. If you have a question, I know this woman, Rhea Foreman, who is excellent at them. But here you can see we did not have that available. So there's some evidence here for this validation, but we did need this to kind of incorporate the genomic stress index into the trait model. So this is just showing the final results. So when we use the NNP stress index into that trait model, we found P to C, POC to POP agrees well with the predictions based on phosphate alone. So the phosphate model by itself is used, is seen in the red line, and the trait gene model you can see in the blue, and there's good agreement in the Atlantic Ocean, which we expect a lot of the initial observations are based in the Atlantic Ocean. For the Pacific, we also see a tight relationship between the predicted C to P based on phosphate alone, and also for the trait gene model. Now, where we see an improvement in the model is when we incorporate this, this nitrogen stress index. Because again, it's now the growth isn't just diffusion limited, but we also have this transporter limitation as well, where if the cell doesn't have enough nitrogen to invest in the nutrient uptake transporters for phosphorus, then it can't draw down, it can't uptake the residual phosphate, resulting in more phosphate in the surface water. Sorry, so as that nitrogen limitation kind of takes hold, there's kind of the secondary hold on uptaking that extra ambient phosphate, resulting in an increase in C to P. That's our theory for why we see this relationship here. So just kind of, I'm not going to go much into this, but we also went out to the Pacific Ocean, 2016, 2017. So this was a transect from San Diego all the way down to the Antarctic ice shelf. And again, what we saw is this is the South Pacific subtropical gyre, and this is the the edge of the North Pacific subtropical gyre. And we saw elevated C to P slightly in the North Pacific region, but much more so in the South Pacific subtropical gyre. But when we see the phosphate concentrations at this transect, so phosphate is in blue. What we see here is in the northern portion, it's a little bit hard to see, but the phosphate concentrations don't differ too much between these two regions. But what we do see is we see a very strong spike in the POC to POP in the South Pacific subtropical gyre. And when we use the original trait model, or the single nutrient model with either nitrogen or phosphorus to try to predict the ratios of C to N, C to P, and N to P, we see a very large residual. Just a very big mismatch between the predicted ratios versus what we observed along this transect. So again, we did not incorporate the gene model yet, but I'm kind of looking forward to, this study came out this year, Usteik Larkin et al., where we just took this kind of index. It's slightly different than the one I was showing you before in that model. So to look at this one, you want to go to this study to look at the exact differences. But in general, we see this global pattern of nutrient limitation that is not just focused on nitrogen, but also phosphorus and co-limitation with iron as well in certain areas. So the observations that we had from the bottle incubation experiments and that study by Twining et al., we can see this shift in different limitation patterns in this Indian Ocean transect, as well in other transects in the Pacific, Atlantic, and Indian Ocean. So there's really just a lot of potential for this kind of analysis where when nutrients aren't necessarily available, or we want to summarize kind of all these different nutrients that aren't necessarily measured, including dissolved organic phosphorus or nitrogen, this genomic nutrient stress index might be an alternative way to incorporate into these biogeochemical models. And this is just showing some of what that looks like, in the Pacific Ocean, where again, we have this shift between along this transect where we saw, so this where I'm highlighting here, where we saw the very high C to P and C to N values is where we see evidence for this nitrogen stress again. So again, we may be seeing the same kind of hypothesis where we have severe nitrogen limitation in the South Pacific Gyre, leading to really high relative ambient phosphate turnover. There was a study by the Outpace crews, I think Thierry Mouden et al., who looked at phosphate turnover rates, and they were on the order of days and days in this gyre. So something is really limiting phosphate uptake in this region, and we hypothesize that it's due to nitrogen limitation of these nutrient uptake transporters. So just, I wanted to end here just to show the potential for this kind of analysis for kind of solving these kind of puzzles that we're finding as we're going to new areas and collecting new samples. And I just wanted to acknowledge just all the collaborative effort on this study. In particular, I wanted to highlight for the metagenomic samples and just developing this index, especially Elise Larkin and Lucas Ustic. Elise put a lot of work into just developing this whole protocol, and Lucas with just developing this nutrient stress index. And I also wanted to especially call out George Hagstrom. George Hagstrom, I think codes faster than I speak. So if you have questions about the trait model, please talk to George Hagstrom. And if you have questions about this metagenomic analysis, please talk to either Elise Larkin or Lucas Ustic. And just wanted to thank everybody from the Martini Lab at UC Irvine, the Princeton group, and also the Bigelow Laboratory for Ocean Sciences, and of course the crew and the science parties on each of these cruises, and my funding sources as well. So in that note, I think that's it. So I have time for questions now. Thank you. And here.
If anybody does have questions, feel free to jump right in and ask, unmute yourself.
Amazing
Or you
job,
can-
Cathy.
Thank you.
So your title, more genomic markers for the nutrient. So it's really cool when you have undetectable these nutrients and you still pick up these genomic markers. I was wondering, doing metagenomics or metatranscriptomics is an excellent way to get these gene expressions. But also it's very intensive, bioinformatic work, and somebody needs to do this kind of level of work for every sample may not be feasible. Did you come up with a list of genes where they could just, use the genetic markers on qPCR or something, a simple thing? Could we easily tell how the indication is, or nothing but like a genetic markers?
Yeah, there is so the nice thing about that newer study that came out, they actually divided the genes into medium stress and high stress for iron, phosphorus, and nitrogen. So that I think the supplementary material has those specific genes and also their location within each of these reference genomes, to make it so if you wanted to streamline this analysis to a few core genes instead, that maybe show the most variation among these transects, that it's definitely a possibility. And I encourage you to talk to Lucas and Elise about that as well.
Yeah, I was sure. We work on the Great Lakes area, on the Lake Erie. We do most of the time this phosphorus and nitrogen overflow in the lake and the cyanobacterial blooms. So,
yeah, good luck with that.
Yeah.
We do have a question here from Puni. He says, fascinating study. I'm curious if you guys measured salinity in the transect in your Garcia 2018 paper.
Yes, we did. This was part of the Go Ship. So this series of Go Ship cruises it's Go Ship now. It was WOS and I forget. They're a decadal transect where they measure all the hydrography, all the physical ocean parameters from the surface down to basically the down to the deep ocean. So they measured salinity all along the way. Yes. Are you wondering if so salinity? Actually, that's a good question. In the Bay of Bengal, I forget the author, but there's a lot of really great work there. Looking at the freshwater inputs to the Bay of Bengal and from a lot of the river output in that area. And a lot of the river output will have very low phosphorus concentrations, which might partially be why we see this low surface phosphate signal going into the Bay of Bengal. But you have this really fresh, warm water in the Bay of Bengal that kind of places like a cap over the deeper water there. So there's this phosphorus limitation that is kind of, the salinity signal might be able to be another way to pull that out, at least in that area, just because it would indicate that freshwater input in that region. Again, that's just a theory, but I know there's other work there's a lot more work that's been done on that region.
He said, thanks if you don't have the chat up.
Oh, yep, I just brought it up.
Okay.
I'm still not sure how the screen share works. Do you guys just see my PowerPoint or everything?
Yeah, we're good. We don't see your chat or anything.
Oh, okay, yeah. I was worried like every time somebody pops in the meeting, you would see them straight across. Okay. Yeah, thank you everybody for popping in as well.
I
ask a
oh, okay, go.
Can I ask a question that it's just to be an advocate for Satan? You know, it's an awesome study, and the things you're doing are pretty amazing, but it almost strikes me that it feels like the questions could be bigger than what they are, you know? So like back in the day, we answered these kind of questions by doing bioassays. And you're essentially using genomics now to approach that question and tell us you know, something about nutrient limitation and, you know, mostly things about nutrient limitation. But I feel like there's so much more you could do with those data. And you probably are, but I'm just curious, you know, I feel like these questions, you know, limnologists and oceanographers, we're so focused on nutrient limitation, and I feel like it's almost time to get over it. But that's, yeah.
That's why I kind of pulled out that study way at the beginning that showed you think we'd be over it by now. Let me see if I can.
Well, in my defense, I came in late, so sorry if I missed that.
Oh, no worries. Anyway, I showed this this output of some of the model So with these different environmental drivers in the ocean on net primary productivity, the classic ones we've been studying for decades, light, nutrients, temperature, there's still really wide disagreement on what is the effect of nutrients on primary production, net primary productivity among all these different CESM models. So it is actually quite an intensive, to do it the more traditional ways, doing the bottle incubations on board, spiking them with nutrients, running those dilution experiments. It is still very manpower intensive and expensive for ship time. So in a way, the sequencing, of course, is expensive, but you can collect these very high resolution samples and go back to them later and kind of take out the influence of kind of like the in situ bottle methods that you might also be considering. And I know most of the bottle incubations, I feel like nitrogen stress always comes up, and it might be harder to see these co-limitation factors. So even though we keep on studying nutrient stress, I still think we got a lot to learn because we don't have it nailed pat on even when it's more important than temperature per se going forward.
No, I agree. The co-limitation issue is complicated and really important, and we're seeing evidence for that in lots of places. And so, yeah.
Yeah, we chose so this is kind of a good case. This is a good analysis to show that this method works for, in particular, whatever the major stress gradient is in your ecosystem. So if that might be water potential, then potentially you could do a similar analysis looking at gene gain and loss, or developing a similar index if it's not so much gene gaining loss, but using metatranscriptomes or proteomics instead. So just kind of like this type of method of looking at a gradient of stress that shows adaptation among these different cells. For us, it was nutrient limitation appeared to be the strongest gradient. But if you step back into a different ecosystem, you might see another one that this analysis might just be a template for.
I have a question for you, Cathy. So you focus mainly on like the picocyanos, and because you said that they're streamlined genomes, do some of the more larger phytoplankton or dianos, whatever you want to call them, do they also drop genes, or are you only seeing this in more of like the picocyanos?
I actually don't know the full answer to that. I haven't studied them myself. That's why I'm not really sure. But that is a great question, and one I've really been curious about. How far up the scale of phytoplankton size classes can you go before this analysis becomes too noisy? With Synechococcus, we definitely show it still works in this Garcia et al. 2020 study. But would it work for pico-eukaryotes? Would it work for diatoms, coccolithophores? That's a very good question. I'm sure there are people out there who know the answer, and I will probably go straight and look for it right after this. Yeah. But that's when maybe you want to shift towards, instead of metagenomes, maybe developing an index using metatranscriptomics instead. If they have multiple copies, it might be more a question of expression levels.
That's exactly what I was wondering. Yeah, how big can you go until you have to switch to the transcriptome?
Yeah, great question. Somebody should do like a detailed analysis of that.
Any other questions?
I hope you guys laughed at my cats a little bit. I got one more. Yeah. I'm a crazy cat lady as well, if you don't know that already.
There was a chat, this was a chat question from Paul, and I'll let you take a shot at answering it, but and people are leaving here too, but any thoughts about light